Good morning, viewer. Welcome to our Glory Globe TV. Here's a breaking and most amazing news for our viewers. Yes, in our bodies, millions of cells meet their end on a daily basis. Contrary to popular belief, cells don't just explode when they die. Instead, a particular protein acts as a trigger for the rupture of the cell membrane. Scientists from the University of Basel have recently been able to elucidate the exact mechanism at the atomic level. Their findings are published in the journal Nature. The elimination of cells is a vital process for all living organisms. When cells becomes damaged or infected with viruses or bacteria, they initiate an internal self-destruct sequence. This essential mechanism wards off the potential growth of tumors and prevents the spread of harmful pathogens throughout the body. Until recently, it was assumed that cells simply burst and die at the end of their life. Now, researchers at the Biogentrum of the University of Basel, the University of Lausanne, and the Department of Biosystem Science and Engineering at ETH Zurich have provided new insights into the final step of cell death. In the scientific journal Nature, they describe how a protein called ninzurin-1 assembles into filaments that work like a zipper and open the cell membrane, thus leading to the disintegration of the cell. The new insights are an important milestones in the understanding of cell death. Various signals such as bacterial components trigger the cell death machinery. At the final stage of this process, the cell's protective membranes is compromised by tiny pores which allow ions to stream into the cell. The common understanding was that the cell then swells until it finally bursts due to increasing osmotic pressure, explains Professor Sebastian Hiller, who heads a research group at the Biogentrum University of Basel. We are now resolving how the cells really rupture. Instead of bursting like a balloon, the protein ninzurin-1 provides a breaking point in the cell membrane, causing rupture at specific sites. Using advanced techniques such as highly sensitive microscopes and NMR spectroscopy, the scientists have been able to elucidate the mechanism by which ninzurin-1 induces membrane rupture at the level of individual atoms. Ninzurin-1 is a small protein embedded in the cell membrane. Upon receiving the suicide command, two ninzurin-1 proteins initially cluster together and drive a wedge into the membrane, explains Maurice Degan, first author of the study and PhD student at the PhD School for the Swiss Nanoscience Institute. Large lessons and holes are formed by many further proteins attaching to the initial wedge. In this way, the cell membrane is cleaved upon piece by piece until the cell disintegrates completely. Behind the report, Glory Globe TV, Delhi, India.